I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them. What do you know about MS?
What do I know about it? It's a very terrible disease. It affects more women than men, I think.
I know it can cause problems with like muscle movement and the nervous system.
I know that it's multiple sclerosis, and that's about it.
I don't know anything. I've heard of it.
But the B cells attack the myelin sheaths, and it causes a lot of damage and mobility issues for the patients.
There's still this problem of ongoing progression of disease. That's an unmet need for patients and an area that we have a lot of research interest.
Hi, everybody. Welcome to our show. Today, I'm very excited to talk about a topic that we have covered before in greater detail. We're going to talk about multiple sclerosis. You'll remember back in Season 4, Episode 2, during the COVID years when we were recording with masks on, we talked with Anne Herman and Tracy Yen about the definition of what MS, multiple sclerosis, is, what are the lesions of the disease, what happens to the neurons with the demyelination. And today we're going to dig in a bit deeper and talk about the breakthroughs in treatment for this condition. With me today is Sherman Jia, who is an expert in the treatment and research around multiple sclerosis. Welcome, Sherman.
Thanks for having me.
Thanks for being here. So Season 7 is about breakthroughs. So you have a unique perspective in that you're a scientist and a clinician who both researches this disease and treats patients. Could you tell us what has been the biggest breakthrough in MS treatment?
Yeah, so multiple sclerosis is an immune-mediated disorder that affects the central nervous system and affects around 1 in 1,000 individuals, close to 1 million individuals in the United States. It's the leading cause of adult-onset neurologic disability. Patients experience multiple sclerosis in many different ways, from fatigue to motor disability, and eventually losing their ability to potentially walk or work and really interact with society. Fortunately, in the past 20 to 30 years, we've made groundbreaking advancements in therapies. So, approximately 30 years ago, in the 1990s, the first approved therapies for MS were made available. These were injectable therapies, interferons, glatiramer acetate, that were really trying to reduce the relapses in acute disease activity in multiple sclerosis. Fast forward about 10 years, in the 2000s, there was another advancement. One of the first high-efficacy therapies were approved. This is an IV infusion that works by blocking lymphocytes or immune cells from entering the brain and the central nervous system. Highly effective for reducing disease activity, but there were still some challenges with safety. The main breakthrough came in with the advancement of B-cell depleting therapies, namely anti-CD20 therapies that targeted a whole different biology, B cells, in multiple sclerosis. And since then, the patient experience has been transformed from a quality of life perspective.
I remember when I was a kid, one of my friend's parents had multiple sclerosis. This was in the 80s, and they were wheelchair bound. They were very disabled and sick and not very old, right? So that's really interesting in that context, that the outlook has improved pretty dramatically over the past 30 years. Can we just review how the patients experience the disease now?
So multiple sclerosis patients have onset of their disease in early adulthood, in their 20s or 30s. And the older patients that I've seen in my clinic who didn't have access to the latest treatments have unfortunately accrued meaningful disability, which is quite hard to reverse. And that impacts their quality of life, their ability to work, and to participate meaningfully in society. The younger patients that I've seen who are in clinic after the advancement of B-cell depleting therapies, it's a completely different world. And because of how effective B-cell depletion is, they can forget about their disease for most of the time. So the difference in these two populations and their experience of the disease is really quite night and day.
I have another just curious question, which you don't have to include this, but I just would like to ask you. My understanding is that presentation with MS can be quite difficult to diagnose, right? Because there are multiple strange types of little bit of numbness, like a little funky stuff with your vision, and it can take people a while to get the diagnosis. Is that still true?
Yes. So certainly if a patient presents to a non-MS expert, then it can take quite a while before a diagnosis is given. And the diagnosis is based on a combination of clinical symptoms, including the frequency of their prior clinical attacks, as well as characteristic brain MRI findings in specific regions of the brain, paired with, when in some cases necessary, additional diagnostics, spinal fluid analysis, or comprehensive eye exams. So it's really the combination of the clinical and additional biomarkers that gives us confidence that it's multiple sclerosis and not a mimic. And there are a number of related disorders that clinicians need to rule out through a variety of other tests before giving the diagnosis of multiple sclerosis.
So there's been advances since we last covered this topic. Could you dig in a little bit more about what we've learned about the biology of the disease?
Yeah, so the disease mechanisms are in multiple sclerosis. I think we have a better understanding now compared to before. When we first tried to look at human brains with multiple sclerosis lesions, the first cell type that pathologists found were a combination of T cells and macrophages that were causing inflammation at the site of the acute inflammation. They didn't detect B cells in those lesions for a very long time. And it wasn't until much later that we saw that not only are B cells important, but some of them may be resident in the brain, in the meningeal tissue or other focal aggregates of lymphocytes. That paired with the observation that in the spinal fluid, the fluid that surrounds the brain and spinal cord, there are detectable immunoglobulins that are characteristic for a clinical diagnosis of multiple sclerosis. These are called oligoclonal bands and are an intrathecal synthesis of immunoglobulins that are specific to the central nervous system and not in circulation.
So that was a clue because antibodies come from B cells.
Exactly. And so that was a clue that B cells may be playing a role. And then later on, a lot of investigators following on the B cell clue tried to use B-cell depleting therapies and saw fairly robust efficacy. And it was that set of evidence that led to the hypothesis that if we really try to develop something in MS, it might have breakthrough potential.
Hi, Maria.
Hi, Wellington.
Hey, Maria.
Hi, Karen.
Okay, vocabulary question, intrathecal.
Oh, isn't that a great word? Intrathecal means inside the CNS. I think the thecal sac is the membrane that encloses the spinal cord and brain. And so what we're talking about here is a disease that is of the central nervous system. And intrathecal is usually, you talk about that when you're delivering into or sampling from the cerebrospinal fluid, which is the fluid that our brains and spinal cords live in, because they don't have blood circulating within them.
Maria, I'm curious why it took so long to detect B cells in the brains of these MS patients. Was it because of the T cells and everyone thought that they were the culprit, or is it something about our brain anatomy?
No, I think it's the former, as I understand it. We always thought that all of the evidence and the models pointed to it being a T cell driven disease. And I think while people look back now and you're like, oh, yes, there was some evidence that B cells were involved, but it was always thought to be more of a secondary sort of phenomenon to clean up. And then when the hypothesis was tested using anti-B cell depleting therapies, it became really realized that B cells were important. And I think B cells have just gotten more exciting. They do more things than immunologists originally thought they did over the years. And so this was a really important breakthrough, understanding that they were disease-driving cells in this disease. So what do we know about what's happening to patients whose disease continues to progress, and what's the underlying biology we might go after to correct that?
So absolutely. In patients who are treated with B-cell depletion, over the long term, depending on how you look, most patients do okay, don't have progression for the long run, and most don't need wheelchair on B-cell depleting therapies, which is great. But if you use some more sensitive measures, a combination of clinical measures, not just your kind of standard expanded disability status scale, we see that a greater proportion, not the majority, but a greater proportion of MS patients do progress despite B-cell depleting therapies, suggesting that there's ongoing injury happening under the hood that is contributing to worsening. Now, what are the causes of that still underlying progression? We're still trying to figure it out. It might be that because B-cell depletion gets rid of the acute disease activity so well, but there might be still ongoing low-grade inflammation behind this intact blood-brain barrier. So two phenomena that have been well characterized in MS include the concept of chronic active lesions, existing MS lesions that have a rim of inflammatory activity that are enriched for microglia. And perhaps that is contributing to continuing tissue destruction around existing lesions.
And that's completely independent of B-cell biology. The microglia would be the pathogenic actor in that context, maybe.
It might be the case that microglia are playing a critical role, and we don't see B cells as often. There may be some interactions that we don't have the right tools to sensitively detect those interactions. I think the jury's still out. Yeah. The other component is the aggregates of B cells, T cells in the meninges of MS patients that might contribute to this kind of smoldering, quote unquote, inflammation that is producing inflammatory cytokines and causing local injury at in the gray matter and the cortices of human brains. So that's one big bucket. Just there's still chronic inflammation happening in the brain despite our best treatments.
Maria, what overall strategies are scientists using to unlock the culprits to these complex multifactorial diseases?
Oh, so many things. I would say that starting with the human genetics is always really important. Rare variants, like people who have a mutation in a gene that leads to an incidence of a disease, we're identifying more and more of those now with more and more genomic sequencing being available. You can connect the genetics to cellular protein and RNA expression, to open reading frames. So for an example might be you could use then a sort of AI and data analysis to say, this gene seems to be involved in a disease in this particular population, in cells from this type of the organ that's involved, say the brain. This protein looks like it might be changed or upregulated, and you sort of generate hypotheses from the masses of data that can then be further tested to identify novel pathways and targets to address disease. So we've covered a lot of ground here. We've talked about T cells, we've talked about disease progressions, we've talked about past breakthroughs in therapy, we've talked about inflammation. I want to ask you about breakthroughs. Where are the next breakthroughs going to emerge, do you think?
There are many potential avenues for breakthroughs. One that has been discussed in the field more recently is the thought of targeting both B cells and microglia using a single mechanism that targets a protein that's involved in both cell types called BTK, Bruton's tyrosine kinase. And this class of medications has the potential to have the benefits of modulating B cell biology, like the anti-CD20s, and potentially targeting the microglia, or these other types of immune cells in the brain that potentially drive the chronic active lesions and the smoldering inflammation happening behind that intact blood-brain barrier. I think the jury's still out on how this class of medication might compare to B-cell depletion with anti-CD20 or other modalities, but it's certainly very exciting that we might have a new class on the horizon.
That is exciting. That it's quite fortuitous that this particular kinase is relevant in two different cell types in the disease. I'm aware that there's been recent activity with sort of cell therapies and, full, immune reset for various autoimmune diseases. I wondered, where do you think that has a place, if any, in multiple sclerosis?
Yeah, so cell therapies, including CAR Ts directed against B cells, as one example, is being investigated in certain types of cancers as well as in autoimmune disorders, including multiple sclerosis. And the underlying premise is that if we use another cell, a T cell, that's engineered to recognize specific peptides or proteins on the B cells that are presumably causing injury, then these T cells, when we give it to the patient, can go to the tissue and organs that really matter for the disease. In multiple sclerosis, that's the brain and spinal cord. And there are a few early studies and early trials first evaluating the safety of cellular therapies because it requires immune preconditioning and observation. And we have to understand what is the relative impact compared to more standard existing therapies, including B cells. But it has the potential, if the T cells targeting certain types of B cells really go into the brain and get rid of the pathogenic B cells that are harder to treat with, say, an antibody, then it has the potential to, quote unquote, reset the immune system in a way to allow for the good B cells to eventually come back later when they do come back.
So it's potentially a curative therapy? Is that overselling it?
It's a potential. Right now we don't know whether any bad B cells or pathogenic B cells will return after the initial treatment, and we just need our early trials to kind of give us that early insight.
What about ways to restore any neuronal function or remyelinate any of these axons? How long do these axons hang around without the myelin for you to intervene and get some function back?
Yeah, so remyelination is another hot topic and an important subfield that the MS community is thinking through. So in multiple sclerosis, the immune population attacks myelin, which are kind of fatty protective layers of sheath that oligodendrocytes in the brain produce to protect the axons for the neurons.
I always think of it as like, a wire, right? When you've got
Yes.
that bit of plastic around the outside of it. I mean, that's a very simplistic way of thinking about it.
That's you're totally right. Yeah. And so the idea is, if there's injury, could we prevent further injury, or could we restore some of the prior injury by rewrapping the neuronal axons by somehow stimulating remyelination through oligodendrocytes and oligodendrocyte precursors. It's easier said than done. It's part of the complex biology is that the oligodendrocyte precursors need to differentiate into oligodendrocytes before any additional remyelination or prospective remyelination could occur. But then there is always a balance between what proportion of the oligodendrocyte precursors do you try to differentiate into that lineage, but not deplete the reservoir of the precursors so that you can facilitate remyelination not only in the short term, but in the long run.
So I have just a curious question. I didn't realize this. So it's not as simple as saying, hey, oligodendrocytes that we already have, you guys just kick out more myelin. That's not going to work. You have to induce more oligodendrocytes to the axon in order to get it to remyelinate. That's
right. That's
okay.
Based on our understanding of oligodendrocyte biology.
Yeah.
But taking a step back, the concept of remyelination or neuroprotection is really advanced by recent understanding of genetics. So multiple sclerosis genetics, the risk of multiple sclerosis, that's been well studied from genome-wide association studies. And
what's the concordance with like identical twins or something for MS?
Identical twins have ballpark a 25% concordance, but only perhaps 40% of that is explained by genetics. So we know MS risk genetics very well, and they clearly implicate immune pathways, HLA, and the genes that are identified over...
Similar to type 1 diabetes.
Similar to other autoimmune disorders, that's right. And the more than 200 loci that have been identified really implicate B cells, T cells, monocytes as the main population. Only very recently, there was a large study looking at, within multiple sclerosis patients, is there a genetic component to the severity of the disease? And that study really starts to implicate CNS populations, central nervous system, brain populations.
So how sensitive your individual neuronal environment is to the immune attack, maybe. Yeah.
Yeah, precisely. Yeah. And so we're just starting to scratch the surface of understanding severity biology, and that's bringing us, helping us pay more attention to oligodendrocyte neuronal biology, and in addition to the immune populations that we know are also contributing to progression.
Oh, that sounds like a very promising angle of research.
Yeah, very exciting.
What about other non-neuronal brain cells and their role in MS? We talked about the microglia. Mm-hmm. Is there any other cell types that we need to think about when we're thinking about MS?
Yeah, so another main glial or support cell in the brain and central nervous system is the astrocyte. Okay. So these are probably the most abundant cell type in the brain, and they play another important role in homeostasis, including providing sufficient types of sources of energy for neurons and other populations. They also have a kind of immune phenotype. So they have these foot processes that kind of survey the environment for potential bad actors, inflammation. And in multiple sclerosis, they are one of the earlier responders when there is a kind of oval-contained region of inflammation for these acute lesions. Astrocytes tend to be present in the area surrounding, at the rim of the lesions, and form this gliotic scar that is meant to kind of fence off immune populations from causing more injury. Now that's the astrocytes trying to perform their homeostatic role, but over the long run, it can lead to scar formation and astrogliosis that down the road prevents remyelination. So for each of these populations, including astrocytes, understanding the nuance of the balance between the protective components versus the potentially impairing repair and other homeostatic functions is critical for identifying the future generations of potential treatments.
No, fascinating. One of the other topics that came up when I was looking into MS is this connection with Epstein-Barr virus. I don't know very much about that, so could you elaborate a little bit on what we believe?
So Epstein-Barr virus, EBV, in the past few years, we have really advanced understanding of the role of EBV in MS.
That's mono, right?
EBV causes mononucleosis.
Yeah.
That's right. Over 95% of the general population is infected at some point in their lives. People are usually exposed starting in their teenage years, but could happen really any time. And one of the pivotal studies that were published several years ago looked at a longitudinal military cohort where service members had their blood samples banked over many years, and some of them developed multiple sclerosis. And the investigators were able to go back to look at their blood samples and correlate the onset of EBV seropositivity before the onset of multiple sclerosis diagnosis. And having that seroconversion was associated with something like 30-fold greater risk for developing.
Oh, wow, that's a large difference.
It's a huge difference.
Yeah.
So much so that the field considers EBV a necessary, but maybe perhaps not sufficient component of triggering multiple sclerosis.
So the combination of the genetic background and the exposure to EBV is what probably drives your risk of the disease.
That's right.
Wow.
Yeah. So you can imagine that we're all excited to better understand now what is the role of EBV in MS onset, but also does it play a role in facilitating immune mechanisms that contribute to progression. And maybe one of the factors that makes B-cell depletion so effective is that they're depleting the reservoir for EBV, because that's the main population that may kind of host chronic EBV.
So is there like a link or an obvious protein on the EBV virus that you think might trigger the autoimmunity? Is there any kind of homologies with myelin?
Yes. So there is molecular mimicry between some EBV proteins and some myelin proteins, and that's been demonstrated. But whether that happens in every single patient, that's unclear. And that if it's true, then that has some potential implications for a whole field of immunology directed toward immune tolerance and using various different platforms. But EBV is certainly very exciting to better understand from a just basic biology perspective, but also implications for treatment.
Very interesting. And I guess you could imagine a future where if you have EBV seropositivity, that presumably can be measured relatively easily, and you have MS risk genetics, would you proactively treat somebody to prevent them developing MS? What would be the risk benefit, do you think, in that circumstance?
It's a fascinating idea that many people have thought about, but maybe hard to implement in a trial setting. So part of the challenge is identifying individuals truly at risk.
Yeah.
So examples ideas that folks have discussed include first-degree relatives of MS patients who have a high genetic risk background, who have, in this case, not yet been infected or with EBV. And among those individuals, if you give some kind of treatment, would you be able to prevent the initial onset of MS? I think that's an attractive idea, but implementation is challenging because of the number of patients that you need to recruit to detect that signal.
Yeah. But similar things have been tried in the type 1 diabetes field, right, with first-degree relatives and trying to prevent onset of type 1 diabetes. That's really fascinating. So when I chatted with Danielle, who hosted the last podcast about MS, she was curious to know in the more recently, as we're developing more and more different new modalities for therapeutics, like we talked a little bit about cell therapies, we have gene therapies, siRNA, all sorts of different types of therapies becoming available. Where do you see those playing in the MS space?
That's a great question. There are several factors that go into choice of modality. If we're prioritizing CNS penetrance, then small molecules might be able to do that better than, say, your traditional antibodies. Cell therapies, because everyone's thinking about it, certainly has potential to, in this case, go into the CNS and brain, but there are complexities with respect to manufacturing, cost, and question of which population do you target given the complexities. Fortunately for the patients, that's only getting better. For the new rounds of therapies after B-cell depletion, I think it really depends on if we have a key biology that we know how to target. Let's figure out a way to target it first before, so that patients get the benefit of reducing disability progression before we make advancements on the modality part.
Are there, I was thinking specifically about siRNA, are there specific genes that you would want to do we knock down or modulate in specific cell types within the brain? Have we got that far in the MS world? I'm thinking of parallels to the sort of Alzheimer's and Parkinson's space where those are being explored.
Yeah. So siRNAs has not yet been developed in multiple sclerosis, partially because the existing targets are around immune populations.
Yeah.
And so as we better understand the biology of CNS populations, where we know that the direction that we need to modulate is down, then siRNAs can be a potential approach. The challenge in MS, just like many other disorders, MS is not a monogenic disease. Mm-hmm. So there's not one clear gene that we can downregulate in, say, neurons or oligos that will really drive home the impact on progression.
So if I were to ask you now, 5-10 years from now, if you were to say again, the patients I treat now, it's completely night and day from how it was in 2026, what would that breakthrough look like to you?
A few potential ways. One, if we figure out how to potentially cure the disease, which could mean effectively stopping their worsening and maybe restoring some of their function, that would be a real breakthrough, because right now, even with our best treatments, as we discussed earlier, there's still some worsening of the disease. And a further breakthrough may be if we can figure out how to prevent the onset of multiple sclerosis in the first place, either by targeting potentially one of the main triggers, EBV, or another relevant biology.
Let's hope we get there. If this is true and that there's a viral driver, you could imagine it being almost like cervical cancer, right? Where the majority of the cause is through HPV, and we now have a pretty good vaccine for HPV. So we're imagining a world where nobody needs to develop this disease because we understand the key driving pathology and prevent it would be a true breakthrough. So how can AI help us understand this, help us develop better treatments? We have these wonderful tools now in biology for understanding disease pathobiology. How is that impacting the MS field?
There are two components that I'm super excited about. One is the amount of data that is becoming available that through publications and other sources that are accessible to any researcher. And this includes deep molecular profiling of brain tissue in multiple sclerosis, and in some cases, access to clinical trial data sets with biomarkers. So that's the one set of super exciting data if one can figure out how to access and how to put it together and do the nuances of the statistical analyses. The other component, as you mentioned, is artificial intelligence and machine learning. And I'm still learning about the latest and greatest in AI, but there's hope that emerging tools may pair Genentech computational biologists with these large comprehensive data sets to allow us to do a deep dive faster and more efficiently than ever before, including for folks who are not deeply trained in the computational sciences. And we're thinking through how to leverage both technologies and data sets in our research, as many groups externally are. But there's still a challenge of after combing all the data sets for signatures and associations and genes and pathways, what is the right way to identify a potential treatment? And for me, that's still an art and requires just time and a lot of dedicated, nuanced question and answers to try to eventually make some progress.
The story you told about the discovery of the association of EBV is an interesting one. That was, I guess, a little bit old school, right? A beautiful set of data that had been collected through the VA. Was it through the VA? Which I think has those types of, they're really precious, those types of longitudinal patient samples and clinical data. But I think with our world of all of our health tech and electronic medical records, there's going to be more and more information like that out there to mine. Do you think that could potentially lead to finding new associations between environmental stimuli and the disease?
Yes, and in a way that's already happening.
Yeah.
And those associations each contribute some component, and the challenging part and the art part is figuring out which of those may be the key to unlocking that next therapy. So absolutely real-world cohorts leveraging potentially AI approaches, that's already happening, and we just need to kind of figure out how to distill the key results from potentially some noise.
Yeah. Well, it sounds like a really exciting time in the world of MS research and also new drug development as well. So I look forward to maybe having you back in the podcast in a few years and finding out what we have been able to do for patients with MS in that period of time.
That sounds great.
Maria, awesome episode. There was a lot of information there to unpack. My question to you is, you were excited about a couple of things there. Are you more excited about prevention, or are you more excited about the underlying biology and how we could do treatments in terms of a breakthrough?
So I'm going to answer that actually in that that's not two separate things. I think the data and the availability of it and the use to understand disease pathobiology should be able to do both, right? If we understand what's driving a disease, we should be able to better understand how to stop it ever happening in the first place, and also how to develop better therapies. It's exciting times.
Not to be a party pooper here, but what kind of timeline are we talking about for the next MS breakthrough?
Gosh, I wouldn't like to, make any particular predictions, but I can say that the sort of with the quantity of data that's being generated and the ability of AI to help us sort through it, an individual scientist can now work through ideas and refine what hypotheses to go and actually test in the lab much more effectively than we were able to do even a couple of years ago. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It will help new people to find us. And be sure to subscribe. If you have a question about the show, you can contact us at podcast@gene.com. That's GENE.com. And now for me, it's back to wrestling with data.